calcium is the most important, okay, what really regulates. If you see the skeletal muscle, you remember it was what do the skeletal muscle, good, what about skeletal muscle? The skeletal muscle plasma membrane has received what? Acetylcholine. As a result of acetylcholine receptor has opened. What kind of acetylcholine receptor? Nicotinic type of. As a result of that, sodium ions have entered. As a result of that, the membrane has depolarized. As a result of that, calcium spike is there. Am I okay so far? Here the sodium has relatively less important. The boss is calcium. Okay. Anything happens, I mean, if the message arrives from outside, from the cavili, sarcoplasmic reticulum, or even from outside, get calcium and start doing your business. Point number 1. Is it very clear? So, calcium. Here is a relaxed cell, here is a contracted cell. I will directly take you to the last point. Whereas the skeletal system is very efficient, very fast, and starts acts at the level of milliseconds. And it better be because if there is a pinprick and if I have to remove my hand, it should happen in shortest possible time. Okay. But relatively smooth muscle system, comfortable, no rush. What is the rush if peristalsis is delayed by 2 minutes? I am serious, it does not matter. In some places it may, but it is very slow system, okay? Slow system. Everything is slow, the response is slow, then the response will last for a very long time. It will also take for the response to pass away. It is a very slow system, okay? Some are very slow, some are not so slow. But as compared to skeletal muscle, this is essentially a very slow system. The frequency may be 1 to 10 or to 100 to 300 of the skeletal muscle. Are you appreciating the point now? The cross bridges have far less ATP activity as compared to skeletal muscles. Of course, ATP has to be there. What am I talking about? Actin-myosin cycling, cross bridge, hello, and for every cross bridge you are going to need ATP. All that is going to happen, but on a very slow scale. You know why it is slow scale? Let us take okay, I will take one of you and draw 500 ml of blood. Okay. As a result of that, your blood volume has gone down. I am sorry, blood volume has what? Gone down. As a result of that, if the physiological response is not there, the blood pressure is going to fall. Okay. So, how is your system going to respond? Your system is going to respond by angiotensin, okay, and then immediately reduce the diameter, particularly of your large veins. Okay. As a result of that, you reduce the volume of the entire available for the blood, so that the blood pressure still remains, I mean, homeostatic, is still quite constant. Are you okay so far? Now, in that particular stage, the blood vessels which have contracted, they are in no hurry to relax, okay. They will remain contracted for as long as necessary. Are you with me? Till no new blood comes or till you do something, they will remain contracted. Are you with me? I mean, that is not a function that comes and goes. No, that is a function that has come and that has come to stay for some time. The smooth muscle cells have evolved a system in which it works slow, it stays for a very long time. Now, here is the beauty of the situation, and it does so by utilizing a minimum amount of ATP. Are you with me? How it does, I will show you. I am trying to draw a contrast between the skeletal muscle system. Skeletal system is very fast, very efficient, comes and goes quickly, and as long as it comes, as long as it's a factory that keeps on using ATP. Okay, are you with me? And since it uses so much ATP that often the mitochondria cannot supply, therefore you need creatine. You remember the creatine? We did it in this class. Creatine, you need creatine. Okay, smooth muscle system, there is no rush. Okay, and because sometimes you need to sustain the contractions for. Look at the valve. Which valve I am talking about? The valve that is lying between the stomach and the duodenum. Stomach and what? Duodenum. And the food has passed from the stomach after digestion, acidification, it has gone into the duodenum, okay, and it does not want that the food from duodenum, digested chyme from the duodenum should go back. So, the wall closes. If once it closes, it remains closed for 5-10 minutes, okay, right. So, during that period, if the skeletal muscle were to do it, which means for every second I am going to use ATP, smooth muscle cell don't do that. Okay. So, how they do it, I have not answered that question, that is a very important question, I will answer it in due course of time. But I am just tell you that these cells they can sustain contraction for a very long time with minimum utilization of the, okay, look at those to me, yeah. Yeah.
Is removed from the system, like after inducing contraction, they will remain contracted for some period.
They will remain contracted for some period. They will remain contracted for some period. But angiotensin also won't go so quickly. Okay, new angiotensin will keep on coming. I mean angiotensin is not like a neurotransmitter will come and go. No, it is a hormone will go and stay. Okay, one very simple question for you. Don't read anything, just look at the two images and tell me what is the differences between, what difference do you notice. You are not supposed to read, you are only see the image. You are taking too much time, your time is done. Every cell has gap junction and no cell has a gap junction. Okay. Is the difference clear? Whereas most of the smooth muscles belong to this category, that is what we have seen. The occurrence of gap junction like alimentary canal, aorta, okay. There are very few tissues, smooth muscles, okay, where there is no gap junction, okay. And in that particular case, every muscle cell then has its own nerve supply. This is somewhat like a striated muscle or the skeletal muscle. Are you with me so far? And these are particularly, you see this kind of arrangement, what tissue am I author is showing you here? What is this tissue? Here, what is this? You can't see, I will not show you, I don't want you to read. Can't you identify the image? Eye, iris, and the opening in the middle is the pupil. Hello? And these are the smooth muscles. And these are the smooth muscles under autonomous nervous control. What is their function? What is their function? You go out, reduce the aperture, okay, and you go into darkness, okay. You can't tell your iris that you smile. No, it happens autonomically. Autonomous system does it, okay. Then you enter into the dark room, okay, and then that there is less light there is pursued by the certain retinal ganglion cells. They know, oh, light is not enough, okay. That information is taken to the brain stem, and then from the brain stem then that information is taken to the centers of the sympathetic nervous system, then sympathetic nerve supply goes and supplies these muscles, so that under the influence of increase the aperture, okay. Very fine, extremely small, extremely delicate cell, smooth muscles of your iris. Every single cell has a nerve supply, okay. So, as if nervous system can control every cell independent of the other, okay. And this phenomena you find in the iris. You also find, what is goose flesh? Have you ever heard a very nice piece of music and responded by having goose flesh? Goosebumps. Okay. So, that happens because a hair stands on its end, and that happens because there is tiny muscle here. Can you see this muscle here? Very tiny muscle in your hair, okay, that pulls the that pulls the hair papilla, and then the hair stand there. These cells, these muscle cells, also smooth cells, and it's an autonomic response, and these muscle cells are also, okay, of this type, and this type are called as multi-unit smooth muscles. What do you call? Can anybody stand up and tell me why what? A single unit is single unit and multi-unit is multi-unit. Use your common sense and give me the answer.
In the single unit there are gap junctions, so it is kind of connected system.
And they all work in tandem. They all work in coordination to one another. Whereas this is not so. Okay, therefore we will call this as what? So here every cell is a unit. So whole thing is a multi-unit, and here all cells put together is a unit. Therefore single unit. Did you appreciate the argument there?
Excuse me, sir.
Yes.
what will, like, why do you need a multi-unit system in those eyes and stuff? What will happen if there is single unit system in that?
I do not know. I do not know. What I can try to guess is, I have pondered with this question. I think for fine control means I will tell you what, you want a cell to contract only so much. Are you with me? It means you are going out. See how much light is there? There will little more bright sun. Okay, so depending on that, you are going to reduce the aperture. If it is very bright, you are going to reduce the aperture. For that precise control, this is my guesswork. For the precise control, I think nature has thought that this is a better way to do it. I may be daydreaming, I do not know. There is another wonderful phenomena that you find in the smooth muscle system. I will tell you what. I will take a balloon. Hey, listen to this, everybody. I will take a balloon, and in the balloon, I will put 100 ml of water. Okay. Under the pressure of the water, certain pressure is being exerted on the wall of the balloon. Yes or no? Okay. Now, I will put 100 ml more. Okay. So, the pressure has increased. I do not know whether it is double or not, that is a physics problem, I do not know, but it has definitely increased. Okay. It has definitely, so the wall is trying to press the water, okay, and the water is trying to expand the balloon, and it with 100 ml so much, with 200 ml far more. Okay, so far. Listen to this. If my balloon's wall was to be made up of smooth muscles, get this, focus on what I am telling you. If the wall were to be focused of smooth muscles, or instead of balloon I take a urinary bladder, from the slaughterhouse, get urinary bladder, okay. But you can't get it, because for the phenomena you need the nerve supply. So, you just can't get it from slaughterhouse, that won't work. Let's presume. So, you put 100 ml and there is certain stress exerted by the wall on the fluid inside. Okay. Supposing it is x, what is it? It is x, and then I put 100 more, it becomes 2x. Okay, so far. In the case of urinary bladder, it becomes 2x, 1, 2, 3, 4, 5, 10, 15 seconds, it goes back to, it automatically goes back to x. Hello? Automatically goes. Why does it go? I will tell you why, and how can it happen? How does it happen? Let us see. You have, follow the story, it is beautiful, the biology is beautiful. The bladder is here. Okay. Now, you put water in it, it stretches. Okay. The natural phenomena is, once it stretches, it contracts. Once it contracts, okay, there are n number of actin-myosin filaments interacting by way of cross bridges. Are you with me? N number of are doing that. Now, I put 100 ml more, it becomes 2n. Okay. Now, n means what? N number of actin-myosin filaments, we are talking to one another. 2n is much more. Whatever had increased, those actin-myosin filaments, they separate from one another. Hello. So, it was originally it was 1n, with 100 ml it was 1n. Are you with me so far? Yes or no? Tell me. If no, I will explain again. Let us do it again. Put water, constriction. If you stretch it, it will contract. As a result of, why is it happening? Because actin-myosin filaments are interacting with cross bridges. How many cross bridges are there between, if I actually count, how many are there? N. How many are there? N. I put 100 ml more water. How much the number has gone to how much? 2n. Now, but 2n means those cross bridges are more. After 5 minutes, those additional cross bridges will delink, and the number of cross bridges will go back to n. The answer to that question is, you will understand the philosophy behind this. You have been to washroom, the bladder is empty. Are you okay so far? Now, every time the kidney adds a drop and a drop and a drop and a drop, the bladder stretches. Okay? And as the bladder stretches, there is more and more interaction of actin-myosin filaments. Is it a good idea to keep on increasing actin-myosin filaments interaction and keep on utilizing ATP for as long as you go to the next washroom again? It's a bad idea. So, what nature says is, well, the moment there is I will increase it, but I will also delink the actin-myosin filaments. This phenomena is called as stress relaxation of smooth muscles. Takes time to appreciate, but it is an amazing phenomena. Let us see what there is. Author gives an example of stomach. In the stomach, you add food. You are eating food, okay? One chapati, second chapati. So there is load on the stomach, okay? Now, as a result of more food, there is stress, okay? But eventually the relaxation happens. So, is the wall of the stomach pushing the food with the same force with which it came in, which it happened in the beginning? No, no, no. There is a great economy in that. You do the same experiment on the stomach, but at this particular time you take the scissors and cut the vagus. Are you following the experiment? Cut the vagus. And when we cut the vagus, what do you find is, when, okay, so this is the stomach. In the stomach, we are measuring the pressure. How are we measuring the pressure? By way of manometer. In that manometer, I am not using mercury, I am using water. Are you with me? And I keep on giving, and I measure the pressure to be 20 cm of water, 40 cm of water, and this is my manometer. And I find that the food goes on increasing, but even if more food is there, the more or less the stretch on the wall is not really gone up. Does not mean much, but I will repeat the experiment by cutting the vagus, and then what happens? Tell me. It is increasing, means what? As the pressure kept on mounting within the lumen of the stomach, it gave the information to the brain kept on calculating that, and brain kept on sending the information, you relax, you relax, you relax. As a result of that, this pressure this remain constant, but the moment you remove what? The input and output via the vagus. So, the moment you cut brain out of the system, well, the pressure goes up. Are you with me? It is amazing biology, amazing biology. Okay, so this you read about the phenomena of, okay. Now, what we will do is, we will take an electrode. What is this electrode? It is a fine capillary, we have done this. There is a capillary there. In capillary, I am going to fill in with what solution? What solution? I told you once. Strong solution of KCl. What is it? Strong solution of KCl. What is it? Yeah. Strong solution of KCl is the fluid that, because it is a conductor. Okay, that ions are the conductor. I am going to put here in a smooth muscle, and I am going to connect it to the oscilloscope, and I am going to read the. You know what happens? It is very simple. What I find is, even under resting condition, the reading that I get here is, supposing it is minus, it is inside the cell, and I get the voltage of minus 50 millivolts. How much do I get? Minus. It does not remain constant on its own. It goes to about 40. Over a period of how many seconds? Maybe 3-4 seconds, 50 to 40, again 50, 40, 50, 40, 50, it is oscillating, which you do not, this phenomena again you do not get in the skeletal muscle, it is oscillating. Is the cell responding? Not really, it is not really, it just. But if you stimulate the cell, in this particular phenomena, if you stimulate the cell either by stretch or by acetylcholine or by stimulation of parasympathetic nervous system, then you suddenly find that this, which was oscillating between 50 to 40, it starts oscillating between 45 to 35, and then occasionally it gives the spikes, and the spike here is the action potential. So, this is not action potential, this is action potential. And if you stimulate further, then it starts oscillating between 30 to 20, and you can see the frequency of oscillation goes up. Means what? You are stimulating it more and more. As you stimulate it more and more, then the oscillations go towards the 0. Okay. And as it goes towards the 0, at the peak, you see it goes here and then it shoots, and then as you, when you are between say 20, 30 to 20, it has maximum number of and on the other hand, if it goes below 60, 70, then you say that the cell is hyperpolarized or the cell is inhibited. Cell is what? Means the cell will not respond even to the regular stimulatory agents or traditional stimulatory agents. We will talk about it. We are talking about the smooth muscles. Okay. Just see what this cell is responding to. It responds to acetylcholine, parasympathetic stimulation, cold, stretch. If you give this cell is hyperpolarized to epinephrine or sympathetic stimulation. Why am I showing this to you? I am showing this to you because in the case of skeletal muscle, all you could do is give acetylcholine and stimulate the muscle cell. Are you with me? Do you have a means of inhibiting the cell? Yes or no? No, you don't have. Your only tool is to not to give a signal, so that whatever acetylcholine is there, it will be broken away by acetylcholine esterase and your cell won't contract. You have no means of inhibiting a skeletal muscle. Get the point? Do you have no means? You can either stimulate it or not stimulate it. When you say not stimulate it, remove acetylcholine, the muscle cell won't contract. Okay, so far? Are you ready for a little interesting detour of the statement? In the case of insects, they have an inhibited transmitter, and the skeletal muscle in the case of insects can be inhibited also, just as it can be stimulated. It can be stimulated with glutamate and inhibited with GABA. In the case of vertebrates, stimulated with acetylcholine, no inhibition. But smooth muscle can be excited with da 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 and inhibited with da da da. Are you okay so far? So, here is a patient of asthma, and what treatment did we recommend in one of the previous lectures? Immediate, say that again, injection of adrenaline. Injection of what? Adrenaline, addition of adrenaline. What will the injection of adrenaline do? Look at this graph and tell me. Relax. What is it? It will relax. The answer is here. It will relax and it will dilate, and the person will get, the patient will get some relief. Okay. We have done this. You have done this. You are talking about what? Isn't this very familiar? Okay, so this is an branch of the arteriole, and we have those smooth muscles, and these are the capillaries. No smooth muscles here, and these are in the veins, and you can see the smooth muscles there. And okay, this is an examination for you. So this the cells in this capillary blood work are experiencing shortage of oxygen. How will these cells, if they are say of heart or brain, how would they respond? Tell me. Yes, you are right. How? What is the agent that is being secreted? Adenosine. You remember adenosine? Yeah, so then author tells us here that meta-arterioles and preauricular sphincters without nerve fibers can still, I mean there is no nerve connection here. So these nerves, so these smooth muscles are responding to what? Can still respond to by the following factors cause smooth muscle relaxation: adenosine, NO, hello, lack of oxygen, excess of carbon dioxide, increased H ions, increased K ions, diminished K, all those factors can. Contrast this with the skeletal muscle. The poor guy has only one thing, acetylcholine, either relax, either contract or don't contract. That is the end of the story. But here just see how many factors are there to control the okay. Just focus on this slide, then I will conclude my today's talk. That is a myosin filament. You remember that? Can you see the two heads there and the two tails going one around one another in a helix? Okay. And then we have also seen that on the neck. So if that is the head, that is the neck. On the neck are sitting some proteins. Actually, they are light chains. What do you call them as? You will call them as light chains. The two are heavy chains, and there are 4 are the light chains. Now, please remember, these myosin molecules are, it's myosin. Oh, I should tell you. There are 40 different types of myosin molecules have been encountered in vertebrates. Different proteins are, you know how miraculous they are, from one protein to another protein. Okay. To my knowledge, I read about 40 myosins, there may be more, I do not know. Okay. So this is a myosin. This myosin is very different from the myosin 1, 2, 3, 4. There are different ways of classifying myosin proteins. So this myosin is very different from the myosin that you will encounter in the skeletal muscle. Have I made my point? It is a different. Okay. Number 1. So this is the, and this is the light chain. Okay. Now you have stimulated this muscle cell. That muscle cell has that that's the plasma membrane, and let us presume that that's somewhere near the caveoli, and as a result of excitation the L type calcium ion channels done. So that calcium there, what is that type? L type, that's going to open. Okay. As a result of that, calcium is going in. That calcium can act on the sarcoplasmic reticulum, which is also a small store of calcium. That phenomena there means this talking here, maybe even CICR. Excellent. As a result of that, more calcium. Okay. Now this more calcium now, okay. Now let us go back to skeletal muscle. There the calcium ions combine with which protein? Huh?
Calciclustrin.
Rhinotype.
Calciclustrin.
Troponin. They combine with what? Troponin. Troponin is made up of 3 units. One guy talks to calcium, another to tropomyosin, and another to actin. Okay. So calcium goes to troponin. Here it goes to another protein which is called as calmodulin. What do you call it as? It is a dumbbell shaped protein which is thin in the middle and thick at the ends. It can combine with 2 calcium ions there and 2 calcium ions there. So how many calcium ions it will combine with? 4, and then it is activated. Okay. And then that complex, calcium calmodulin complex, okay, that will activate the enzyme called as myosin light chain kinase. What is it called as? MLCK. What does MLCK stand for? Myosin, say that again, light chain kinase. That myosin light chain kinase will phosphorylate this protein. What protein? This MLC, that protein is phosphorylated, that this protein will be phosphorylated. Once it is phosphorylated, it is activated, and once it is activated, okay, on the head, this is a protein. Of course, it is all protein, and that protein is ATPase. Hello, that protein is what? ATPase. It was inactive. Now, under the influence of this enzyme, which is phosphorylated, that ATP is now activated. So it will now start taking energy from ATP. Once it does that, it will bind to the actin filament, and it will go over the process of cross bridge formation. Hello, are you good so far? Okay. And at the same time, there is another enzyme called as phosphatase. That phosphatase, this was phosphorylated. Phosphatase will remove the phosphatase, and it will become inactive, and it will give rise to relaxation. Depending on what information is coming from outside, it can contract, and you can relax the muscle. Okay.
When will phosphatase act?
When will phosphatase? It depends on the incoming information. Okay, as long as the incoming information says you contract, it will. Once that information goes, then phosphatase will get active, and then the whole system will go back to the relaxed state.